All right, so today we're learning about Mendelian genetics. Have you guys heard about that before, or is this your first time? With like Punnett squares and things like that? Okay, I mean, awesome. I'll teach you something. So today, it's okay if you don't know what these words mean yet, but our objectives are, for today is that you guys will be able to set up Punnett squares to complete what's called monohybrid cross problems. And also, I want you guys to be able to understand these words: genotype, phenotype, homozygous, and heterozygous. So if you see those words in our lecture, make sure that you take extra care to write down those definitions and get to know those words. All right? Okay, so just for a little opener, I know you guys did this when you took that survey about your traits. Sorry we never got around to using that, but real quick, I want you guys to figure out, if you cross your hands like this, does your left thumb go over your right, or does your right go over your left?
So like this?
Right goes over my
Right goes over my
Okay, so for me, my left goes over my right. I just want to take a quick survey. Wait, guys. So wait, raise your hand if your left goes over your right. Okay. Wait, Christian, is your left over your right? Okay, so that looks like a little more than half of us. Wait, raise your hand if your right goes over your left. Okay, so I guess I kind of expected that more of us, our left would go over our right. And that's because, interestingly enough, putting your left hand over your right means you have the dominant gene. And if right goes over the left, it means you have the two recessive genes. And what that but guys, here, please hold the side conversation. But what dominant means is, remember, we have two humans are diploid, which means we get one chromosome from our mom and one chromosome from our dad. And what the dominant gene means is that if one of your chromosomes has that gene on it, then that's what's gonna be your trait. So even if you have one gene that codes for left over the right, and one chromosome that codes for right thumb over the left, then because you have one dominant gene, the left thumb over the right is what's gonna actually be your traits. And in order for your right thumb to go over your left thumb, you need two genes that code for right over the left. And we'll go more into that in a little bit later in the lesson. And then this is kind of interesting. You would think that dominant traits are more common in the population just because you only need one chromosome that codes for that to make it your trait. But interestingly enough, having 6 fingers is actually dominant over having 5 fingers. So if you get one chromosome that codes for 6 fingers, then you'll have 6 fingers. It just happens that this chromosome isn't very common in the population at all, so we don't see it.
I know girls 6 fingers.
Oh, really?
I'll just work out. She has extra.
So yeah, it is a real thing. Okay, guys, so real quick, I want you guys to think about this. So this is kind of review from what your with meiosis.
Oh, I know.
Do you know your
3 sources crossed over.
yeah, crossing over is one. No, not the strong. That's a different thing. That has to do with like
There's one of them like so with the eye.
yeah, independent assortment.
Independent assortment and
No, it's not that. It's actually random fertilization, which that just means random sperm to random egg. If you guys would you mind the side conversation? But so this is just this is kind of these 3 sources of genetic variation I'm about to go over. This is what allows for variation in chromosomes and variations in genes. And without these 3 sources, then genetics really wouldn't work like we're about to go over. So just to review those real quick, we have crossing over, which I'm gonna I'm gonna be randomly calling on you guys to answer some questions so I know where you guys are at. So first question, Anaya, do you know what phase of meiosis this happens in? It's not telophase.
Oh, prophase. Oh my God.
Yeah, it's prophase. And then, Aaron, do you know, is it prophase 1 or 2? Yep. So crossing over, special thing that happens in prophase 1 in meiosis, and that's again where the chromosomes, they actually get entangled and exchange genetic material. And that's one of the main reasons why we see so much variation in humans, because every single gamete that gets produced looks a little different. So I'll keep that up there. And then guys, again, for today, you don't need to copy everything down word for word. I just want you guys, like usually good note taking consists of just writing down the main points and something that will be useful for you to review quickly later. And usually copying everything down for word isn't the best. And then also, if I'm going too fast, just please raise your hand. I can go back. All right. So the next source of genetic variation is random fertilization. And this really isn't a process in meiosis. This is just the fact that in our lifetimes, all of us produce tons of different gametes. And usually the average person has 2 kids nowadays. So only 2 of your gametes will actually turn into an embryo in your life out of the thousands that you make. So that's just another source of variation of which one actually will turn into a human being or not. Like what? So you can see, yeah, it's just a random sperm with a random egg. Oh no, you're looking at the notes, right? Yeah, you're fine. Yep, there. Alright. No, it's just, I think having 3 kids is pretty common, but it's just the average in America now is like, the average family has like 2.1 kids.
How do you have 2.1?
Well, it's because it's an average. It's because it's basically, for example, if one person had, if you were comparing the averages of 2 families and one had 2 kids and one had 3 kids, then the average of that would be 2.5. So it's just, all right. Wait, guys, please. Okay, so we have our next up. I know I talked to a lot of you guys about this one-on-one, but the third source is independent assortment. And all this is there's honestly multiple ways to think about this. We'll go into it in a little bit here, but in terms of meiosis, it's basically in metaphase, the chromosomes all line up in the middle, right? And it's actually random how they line up. And we have a perfect example up here. So if we're looking at a cell that has 2 blue chromosomes and 2 purple chromosomes, sometimes both blue chromosomes will line up on the left side, and sometimes a purple and a blue chromosome will line up on the left side. And depending on how they line up actually has a huge effect on what the gametes will look like. As we can see here, if 2 blue line up on the left, we're left with 2 blue in one cell and 2 purples in the other. And then over here, these gametes are a mix of all the colors. And this works the same in our bodies with chromosomes that have different traits and genes. Okay, I'll wait a second. So again, while you guys are writing that down, just these 3 processes are basically a huge reason why that humans are so diverse. And without it, we would all look a lot more similar. All right. Are you good, Lily? Or? Okay. Okay, and so now we're going to actually start getting into Mendelian genetics, but these are just some key points to remember. So DNA and RNA carry genetic information. So when we're talking about genes and alleles, we're just talking about DNA. And that it's basically different chunks of chromosomes, and then chromosomes are just made of DNA. And then the genetic code is shared by all living systems. So basically, how this Mendelian genetics, it applies to humans, but it also applies to pretty much every living organism on Earth. And finally, today we're going to be going over Gregor Mendel. He's the scientist who he was the first one to discover genetics and how we know today. And don't worry about writing this down. We'll go into that in more detail in a second. So Mendel, he was actually a monk, and he basically, it's my understanding that he became a monk just so he could spend more time doing science.
What's a monk?
A monk is just
You shake your head.
a monk is just someone who, it's basically a super religious person who just lives with it, and they basically just focus their life on religion. Yeah, but I guess this monk, I don't know how much free time monks have, but he had enough free time to discover the basic principles of heredity. So
Oh yeah, that might
How the heck do you discover?
yeah, I don't know. Maybe he wasn't a good monk. I don't know. So we'll go into how he discovered it, but he didn't. He kind of has a famous experiment for how he discovered heredity. And but at the organism he used to discover them was pea plants. And the reason why pea plants were a good organism to use is because there's lots of varieties. They were pretty easy to control how they mated, and they reproduced relatively quickly. And the reason why that's good is because he got to see generations super quickly. Because if we were going to do a genetic study on humans, we'd have to wait like 30 years in between generations. And that's not really the best for science. And then I guess I can go into this to now. So actually, I'll say that later. I'll just wait. Are you guys good? Or? Okay, so this is super important to write all these definitions down. So basically, again, humans are what we call diploid, which means we have 2 copies of each chromosome. Again, one from our mom, one from our dad. And how we write those down is just with 2 letters, kind of like this. And capital letters represent the dominant version of a gene, and the little letters and lowercase letters represent the recessive version of that gene. So, if we look at right here, this person, we call it homozygous dominant because homo means same, and they have the same 2 alleles, which are dominant because they're capital letters. And then next, and same thing for homozygous recessive, that just means we have 2 recessive alleles, which are represented by 2 lowercase letters. And heterozygous means they have 1 of each allele, and that's just represented by 1 uppercase and 1 lowercase.
Yep.
And next there's 2 types of ways we kind of talk about genetics. We have genotype, and that genotype just refers to the actual alleles that you have in your body. So, for example, the genotype of this person right here would just be a capital A, lowercase a, because that just refers to exactly what their genetic makeup looks like in their body. But phenotype's a little different. Phenotype refers to just kind of the organism's appearance and actually how the traits are expressed. So, for example, because this organism has a dominant allele in it, we know that it will express the dominant trait. And same with this guy right here, we know that will express a dominant trait. So we would say these 2 have the same phenotype, but they don't have the same genotype because one has a recessive allele in it and one doesn't. So that's an example where they can have the same phenotype and the same genotype. Any questions on that, or hopefully, okay? I'll give you guys like 10 more seconds. All right. Is everyone good? Oh, the bottom? Oh, it says phenotype, and then an organism's appearance, which is determined by the genotype. Alright, so. Alright, guys, please put your phones away. Okay, so this is the next thing. So I've been using the word allele a lot. And what an allele actually means in more detail is it's basically just, it refers to an alternate version of a gene. So what you can see here is, this is kind of an example. These are some chromosomes from a pea plant. And as you can see, this part of the chromosome codes for its height. So we have this chromosome codes for tall, and this one codes for tall as well. But sometimes there's slight variations in genes, and that's what we call alleles. So for example, in parent 1, it has the wrinkled version of a gene, but this chromosome has the smooth version of the gene. So that's what we call alleles. Are basically, it's the same gene, but just a slight variation. And that can result in different changes in phenotype. Put your phone away. So, are we good on alleles?
What is somatic cell growth?
So, I mean, this is more with the past unit, but somatic just means like our body cells, like our number. Like, so all it's saying is our body cells have the normal amount of chromosomes, but then sex cells have half the amount, right? That's all. So basically the distinction here is sex cells are going to be haploid and only have one copy of each chromosome. So as long as you're good on that. All right. All right, guys, so now we're going to get into Mendel's experience, like the experiment of how he discovered all this stuff about genetics. And he looked at two different traits, color and seed shape. And interestingly enough, so Mendelian genetics is the simplest form of genetics out there. And again, it just has to do with recessive and dominant genes. But as it turns out, genetics isn't always that simple. Like even things like, I think our hair color doesn't follow the Mendelian genetics path. Like there's like multiple genes that contribute to our hair color. So, but Mendel got kind of lucky. He happened to pick two traits that are coded for quite simply. So that kind of allowed him to figure out the first step in understanding genetics. But again, not every trait is coded how we're about to learn how they're coded. So, his first step in his experiment was to do true breeding between some pea plants. And that means he needed to figure out some purple pea plants that will only produce purple pea plants when they self-pollinate. And that ensures that only genes that code for purple are in the plant. So you know you have a homozygous organism. If all a plant can make is copies of itself, you know that it has it's homozygous in its genes. So I'm gonna illustrate for that for you. So guys, please look over here. So I know I've been using a lot of words that maybe you guys don't understand, but I think this is where it's all gonna start coming together. So in Mendel's experience, he started off with true breeders of purple colored plants. And that means true breeders again means that those purple pea pods, when they bred with themselves, all they produced were more purple flowers. So after you do that, you're left with homozygous dominant plants, which have two alleles that code for the color purple. And he also did the same thing with white flowers. So he got a bunch of pea plants that would breed with themselves and only produce plants that have white flowers. And that's actually the recessive trait. So what he did is he actually, once he had his true breeders, he crossed them and bred them together. And how we determine crosses, it's something called a Punnett square. Again, wait, raise your hand if you've done Punnett squares before. Okay. That's good. You guys can help me teach everyone.
But it's good. I didn't know it was called.
Yeah, no, that's the official name. Wait. Yeah, no, it's all good. If you've done it before, that's more important. Okay, so. Oh. Okay, so again, when you're in sexual reproduction, you get one chromosome from your mom, one chromosome from your dad, right? Or in this case, we can do one allele from our dad and one allele from our mom. So that means if we're looking at an offspring's randomly going to get one allele from the dad or this plant, and then one allele from this plant. So there's basically 4 different possibilities that can happen, and we'll draw those out here. So it might be hard to get my point across because no matter what happens, each parent is going to give the same chromosome. But if we sublabeled them, so even though each allele is the same thing because they're true breeders, if we kind of sublabel them chromosome 1, chromosome 2, or allele 1, allele 2, you can see that we have 4 distinct possibilities. So you can either get allele 1 or allele 2. Oh, thank you. So, does that make sense how Punnett squares work? That you get one allele from your mom, one from your dad, and then there's 4 different ways. There's 4 different outcomes that can happen. So, where did the okay, but so guys, oh here. So in Mendel's experiment, he actually labeled the generations, and this cross right here was called a cross between the P generations or the parental generation. So after we had the violet true breeder and the white true breeder cross, we were left with this, and we'll write the result. So every single offspring that came from the parental cross was big P, little p. And I'm gonna ask someone a question real quick. Right, Owen. So we have big P, little p. Remember, big P is purple and little p is white. So what color would this plant be? So in this case, there's actually a special case called that. Yeah, it's just purple. So this is point, if you have one dominant allele, which in this case is big P, which is, then it's gonna, that trait is gonna show. And there's actually a special case where they do mix, and it's called codominance. I'll label this real quick. Okay, so I want you guys to try to predict what the next generation would look like. So if we crossed two plants that had the big P, little p, genetic makeup, what would the next generation look like? So I'll help set it up for you. So at first, three purple. That's it. I like that. So yeah, I guess Owen. But, so, I don't know if you guys heard of Owen, but yeah, he's definitely on the right path. But right now on your notes, I want you guys to do a practice cross. So just fill in this Punnett square on your notes. And again, it's kind of hard to tell the difference between a capital P and a lowercase p, but these two are your uppercase P's, and these two are your lowercase. So go ahead and fill that in. And remember that uppercase means dominant, and lowercase means recessive. And if you're done, go ahead and talk to the people around you to see if you got the same answer or not. I think some of the flies from the frog cage got out.
It's right there.
Oh, I thought I got him. Okay, guys, but keep focusing on this. All right, let me call on someone. So, Scarlett, did you do the Punnett square? No, that's okay. So Jocelyn, wait, guys, let me get your attention again. So, Joss, what did you put for the Punnett square? So what did you put right here? Okay. And what did you put right here? Good job. All right, guys. Please limit the side conversation. And what did you put right here? Yep. Awesome. So yeah, all you're doing is just kind of filling it out by matching up the columns and rows. And then what goes right here? Yep.
Do you want us to show the percentage of each geometry? Or is that?
You don't have to. Well, you don't have to do it right now, but there will be problems in the future where you have to do that. So, Addison, you tell me if this is two lowercase p's, what would the phenotype of this offspring? And then what color is that? Yeah. And then what would this one be? Purple. Yeah. So if we count, this one would be purple too. That one would be purple too, and then only one of the offspring would be white. So we have 3 purple and 1 white. Does everyone understand that?
So it would be purple?
Well, so again, there's 4 different outcomes that could happen, and we're not sure which one would happen. So basically, we can think about it. We have 3 outcomes where it's purple. So we have a 3/4 chance of it being purple. And then we have one outcome where it's white, so we have a 1/4 chance of being white. So this Punnett square, it only kind of tells you the probability, what the chances are of things happening. But you are right. If these 2 plants did cross, it would be the most likely result that it would be purple. And then again, this is kind of just a quick review of the law of segregation. All it states is that you get one allele from your mom, one allele from your dad. So if this is, we can just say this is like your dad right here, you're only getting one of his alleles. You might get the dominant one, you might get the recessive one. And then the reason why you only get one allele is because those alleles actually get separated during meiosis. Because remember, the end result of meiosis is 4 cells that have half the amount of chromosomes as normal, which is why there's only 1 copy in there instead of 2, like normal. Yep. And then I have a quick question for Asher. Do you know, how would you describe this genotype right here? If you were going to use the words homozygous or heterozygous? Yep, so yeah, heterozygous. So does everyone list, so yeah, if we have 2 different, that's heterozygous. And 2 of the same is homozygous. And what's the genotype of the parents? And you want to label what your letters mean. But that'll make more sense as we go. So I'm gonna do 2 examples for you, then I'm gonna have you guys do some, and then we'll talk about them as a class. Okay guys, so this practice problem says purple, capital P is dominant to white, lowercase p flowers. In a homozygous dominant purple plant, what gametes would be produced? What about a plant that is heterozygous? So this problem isn't asking for a Punnett square. This is kind of a more basic problem than that. But first we do want to write down our genotypes and label our what our alleles are. So again, we have capital P is equal to purple. Lowercase p is equal to white. And now we want to know we want to figure out what genotypes for the parents they're talking about. We have homozygous dominant, which homozygous dominant is just 2 dominant alleles. So that's 2 big P's. And then the next one is heterozygous, which is 1 dominant allele, 1 recessive allele. And now it's asking, what about what gametes would be produced? So this is just asking what alleles these parents could potentially give. So a homozygous dominant parent, it can only give a dominant allele because that's all it has. So it would only produce dominant gametes with dominant alleles in it. And then we have this parent here is heterozygous, which means it has 1 dominant allele, 1 recessive. So it could actually give either the recessive one or the dominant allele. It's all labels. are the gametes that can be made from parents. All right. Any questions on that? Yeah, a lot of this is just knowing what the vocabulary words mean. And it seems like you guys are doing a good job with that. Okay. So, am I good to erase this, or does anyone want to write more now? Okay. So yeah, that wasn't so much a Punnett square problem, but we did need to know what all those vocab words mean to answer the question. Sorry. All right, next is a Punnett square problem. So round seed shape is dominant to wrinkle seed shape. So first we want to write that down. And again, we can choose any letter for our alleles. I'm just going to choose R because the first thing I saw was round seed. You could choose any letter again. So we're going to do R is dominant, which equals wrinkle. And again, this seems like a tedious step, but this is super important because the people need to know what your Punnett square actually means. So now it says a plant that is heterozygous for the round seed shape is crossed with a plant that is homozygous for a round seed shape. Oh, sorry, actually, the dominant is round. I apologize. And little r is wrinkled. So yeah, I made a mistake there, but want to make sure you read those carefully so that doesn't happen to you. But we have, so now that we're labeled, we have dominant is round and recessive is wrinkled. Then let's see what genotypes the problem tells us. So we have a plant that is heterozygous for the round seed shape is crossed with a plant which is homozygous for the round seed shape. Kaylee, do you know what does heterozygous mean? What's the genotype for someone who's heterozygous? Yeah, that's true. So what would it be if these are your allele options? Well, you said it's one of each, right? So it's one big R or one little r? Does that make sense? Because hetero means different, so that means you have different alleles. So it's one big, one little. Does that make sense? Okay. And then Richard, what would the homozygous for a round seed shape, what would that be?
That'd be the same, like if
And then if it's homozygous for the round seed shape, would that be the capital R or the lowercase r? Yeah, so homozygous for round seed shape is 2 capital R's because capital R codes for round. So those are our genotypes. So now the question is asking what % of the offspring will be homozygous dominant. So in order to figure that out, we need to do a Punnett square. We kind of just set up our Punnett square just like we did before. Okay, so it's asking for what % will be homozygous dominant. And if we count, we have 2 of them are homozygous dominant because 2 of them have 2 dominant alleles. So our answer would be 1/2. Because, well, first off, we have 2 out of 4 of them are homozygous dominant. We'll simplify that to 1/2. All right, guys, any questions on that, or?
They're gonna take the air off.
Any questions, or are you guys good? Okay. All right, guys, now it's your turn to try some problems. We'll go over them. Of course I can. Okay, guys, right now on your own sheet of paper, I want you guys to try this practice problem. So I'll give you about, let's say 3 minutes to do it, and then we'll talk about it as a class. And remember, step one, you always want to label your alleles, write down your genotypes, and then do the Punnett square. Those are kind of your 3 key steps, and they're all up here. Sure, I mean, yeah, if you don't know what to do, yeah, that's fine. But here's kind of an example up here. If this, I didn't label it, but if that helps you. Guys, we're running out of time quicker than I thought. So I'll give you guys 1 more minute and then I'll call on someone. Okay, I think hopefully you guys had enough time. Addison, what was your answer? 0? Yeah, it is 100%. So what was your amount of value of Punnett square?
Oh, I didn't pick on
Yeah, no, that is true, but these are capital, which means they're dominant. So every offspring will get 1 dominant allele. You got 100? Okay. So guys, real quick, I'm not, just because we're short on time, I won't label it, but real quick. If we crossed, guys, please listen, if we crossed a heterozygous dominant with a homozygous recessive, then we would get that. And as you can see, all the different outcomes have 1 dominant allele in them, so 100% of them would be tall.
Makes sense. That's what I had.
Okay, awesome. All right, guys, and this is your other practice problem. Please do this quickly. True breeding is homozygous, yeah. So did you say 40%? So there's only 4 omnis, so it can either be 0, 20. It has to go with percentage to go with.
So 0, 25, 50, or 100?
Yes, or 75 too.
I'm a lean towards.
remember, true breeding means homozygous. So,
One chance you had.
Okay, why don't you just do it?
You don't know the need for the example, Lily.
Well, then why don't you ask? Why are you yelling at me?
I see what you're finding when you're back here.
so
Like I said, back here.
should we? So yeah. Yeah, no. Exactly. Okay, guys, I think class is about to be over, so I'll do this real quick. Yes.
How do you go?
Wait, guys, let me get your attention real quick. So guys, so we have a true breeding short-haired cat, which is short. It says short hair is dominant to long hair. So I'm gonna use the letter H. So we have, that's dominant, we have homozygous dominant. And then the other one's heterozygous, which is one recessive, one dominant. And then if we cross those, so as you can see, guys, every offspring has a dominant H, so that means 0% without long hair, because long hair is the recessive trait. Actually, do you mind if I take a quick picture of those? And then,
You see the little conference, the one.
all right, guys. So today that you're going to be completing this worksheet. It has 10 Punnett square practice problems on it, and this will be due at the end of class. But you definitely will have enough time to do it. Punnett square problems don't take that much time. But I'm gonna do one to get with you guys as a class in a second, just to go over it. But before I do that, I just want to review some terms with you guys. So do you guys remember remember there's basically 3 genotypes that you can be in these practice problems. We have a homozygous dominant, which means you have 2 dominant alleles. And do you guys remember what a dominant allele means? Like it. Yeah. So if you have. Yeah. Okay. Yeah. So everyone knows that if you have 1 dominant allele in your genotype, then that is the trait that's going to show up. For example, right here we have 2. This is a heterozygous pea plant right here, and it has 1 dominant allele and 1 recessive allele. But because it has 1 dominant allele, the dominant trait's going to show, which is yellow. And we also have, and the only way the recessive trait shows is if you have 2 recessive alleles, and we call that homozygous recessive. So for this activity, you guys it probably would be helpful to take out your notes if you're not clear on all the definitions. But now I'm gonna walk through the first one with you guys. And does anyone not have a worksheet, or has everyone got one?
Everyone got one.
Awesome.
That's a big workout.
So, you guys just hold the side conversations while I go through this. So I know we got kind of rushed going through the guided practice problems, so that's why I want to do this. Okay, so I'm just going to read the problem quickly. So it says, in humans, brown eyes are dominant over blue eyes. What type of offspring would you expect if you crossed that heterozygous brown-eyed person to a heterozygous brown-eyed person? So we're just crossing 2 heterozygous people. But before we do anything, we need to label our alleles. So because brown eyes is dominant, I'm going to do a big B for brown eyes. I'll just write brown. And then it says blue eyes is our recessive allele because brown is dominant over blue. So I'll do little b blue. Now next step is to set up our cross. And if I have someone, what would the genotype be for a heterozygous person? Does anyone know? Yeah, great, Josh. So it would be a big B and a little b. And we're crossing two big B, little b's. And I'll just make my Punnett square. And now, again, we just fill out the Punnett square like normal. Okay, so then we're left with this Punnett square. So the next question is, what percent of people would have brown eyes, and what percent would have blue? So yeah, so 75% would have brown, and which would be these three, because three of them consist of at least one dominant allele. And then this is the only one where that person would have blue eyes because it's too recessive. So then we could write 75%. Alright. So, yeah, all of these are pretty similar to this problem. You're just breaking down. First, you want to label your alleles, and then you create your Punnett square, and then you want to write the result. So, are there any questions on how to do these Punnett squares, or do you guys feel pretty confident about them? Confident? All right. And okay, so now I'm hoping you guys can do this. I'm sure you can probably do one Punnett square problem a minute if you guys stay focused. So just please work through this worksheet for the rest of the class. But before I let you guys work on that by yourself, I have a few announcements. So, or I guess just one. I want to let you guys know. So because did I say that already? No. Okay, but yeah. So you guys, because we did a lecture over this, we're gonna take off 2 salads off your to-do list. So for the salad portion, please only pick one to do.
Okay, I'll remember that.
So yeah, and then also later in class, I'm gonna have the checklist up here. And I think I was checking so much of your guys' work on Thursday that I may have missed a few of you guys. So please just make sure it's all up to date. Because sadly, I should have wrote it down on a piece of paper so I had a backup, but I forgot to do that. So just make sure you're double checking that. But for now, just go ahead and start working on these practice problems. And I'll be walking around to help.
Drink water.
Oh, then also one more thing I forgot to say is,
Hold your breath with the water.
oh guys, I don't know if I'm sure you noticed this, but this is where you're turning in your work now. This is a 6-period bucket, so when you're done with the worksheet, just put it in there. Yeah. So you want to use the same letter
those
for
are
dominant and
fucking
recessive, but it can be any letter you put. Yeah, exactly. You can, yeah, you usually it's best to pick a letter with a capital. Yep.
Let's be realistic.
Yeah. I'm pretty sure. I think I may have already got you, but I'll chat you.
Please.
All right. but just.
Too spicy. I did.
You might start working on this. We do have any questions? And then, yeah, the example for the first one there.
Was it?
Are you guys feeling good about this? Oh, you're fine.
No.
Yeah, it's honestly not, it's not too bad once you understand it.
No, that was right.
All right, so firstly, over the last couple days, we learned about what it's actually called monohybrid crosses. That's what we were doing, and it's the simplest form of a cross. In the next couple of days, we're going to learn how to do more complicated ones. But our objectives for these past 2 days were, one, students will be able to set up Punnett squares that complete monohybrid cross problems. And if you were able to do the worksheet, which seems like all you were, you completed that objective. And the other objective is I wanted you guys to really learn these four vocabulary words, which is genotype, phenotype, homozygous, and heterozygous. And if you have a really good idea of what those 4 words mean and how they connect to genetics, then you've learned what you needed to learn from this unit. So just a couple questions to go over that. I'll just call in a random person. Lily, what's the difference between genotype and phenotype?
Genotype is the letters and like and phenotype is the actual trait that like it represents.
Yeah, exactly. And then genotype in our Punnett squares model, it is the letters, but those letters represent the actual DNA. So genotype's kind of your DNA and the letters in the model, and then phenotype's your physical trait. So good job. And we got one more question. Oh, Chris was not here. Brandon, so this is a tougher question, but not too bad. So what is the percent chance a flower will be purple if one of the parents is homozygous dominant, given that purple is the dominant trait? So the key is, any ideas? Okay, we'll go over. I can call someone else.
Because half of the square will for sure be purple. But it could be 75, depending on the second trait.
So it's actually 100% will be purple, but I'll show it real quick. So, all right, guys, everyone just look at the back of the board, please. I'm gonna draw it out for you. So this is honestly kind of a trick question, a little bit. It's just, so we're doing a cross. We know one of the parents is homozygous dominant, right? So I'm gonna make H is dominant. I'll put H purple, little h white. So this problem, wait guys, please pay attention. Just this one thing, so the problem told us that one of the parents is homozygous dominant and we don't know what the other parent is. And we're asked what is gonna be the phenotypes of the offspring. So I'm just gonna put question mark here because we don't know the other parent. But we do know that this parent's homozygous dominant, which means that every offspring will get at least one dominant allele, right? And we know that if an offspring has one dominant allele, they're gonna have show the dominant trait, right? Does this make sense?
Yes.
Because if we have one homozygous dominant parent, every offspring is gonna get at least one dominant allele, and we don't really care what the other allele is. So that's why I represented it as a question. All right, so I saw you guys doing the worksheets and think you're doing a great job, so thanks for doing this lesson.